The objectives of this project were to try and unravel some of the unknowns and some of the knowns about environmental flows, and try and show quite clearly the benefits and when disbenefits might actually occur with environmental water. And what's actually happened over the years is as water has been collected and made available for environmental use, it can be applied at different times and in different places. So it really is a managed, almost like a managed flood, to either improve or increase various aspects of the ecosystem. So we broke down the project by looking at what actually happens in the river once environmental water comes back into a river. We looked at what happens when water goes onto a floodplain and how that affects the small animals that might be living or growing in a floodplain wetland system. And we also had a component that looked at how the plants that were growing in the wetland might be affected by those waters.
My component looked at the effect of hypoxic blackwater events on microinvertebrate emergence from floodplain sediments. The project went on for about 12 months.
The first thing that you notice when a floodplain goes underwater is the water changes colour. And it's really the same process as you see in a tea bag. When you put a tea bag into a cup of water, the water goes brown or goes black. And what happens on a floodplain is all that leaf litter and all those twigs and things that are sitting on the surface, they become wet, and the same process happens. All of the materials that come leaching out of that leaf material go into that water, and you get this brown or sometimes even black coloured water. And people know that, and they often see that, and they've referred to that water as black water, but it's part of the process of that floodplain wetting.
Microinvertebrates, we know, are a really important food source for small fish and water birds, and they emerge from floodplain sediments. They have dormant eggs that they emerge from. We essentially went out and collected wetland sediments from 2 floodplain wetlands. So one was on the Gulper Creek floodplain near Echuca, and the other one was on the Edward River floodplain near Deniliquin. We brought the sediments back to the lab, and we basically undertook a laboratory experiment where we put the sediment into glass bottles, shot bottles, and we had 2 experimental treatments. So we had different concentrations of oxygen, so dissolved oxygen, and different concentrations of dissolved organic carbon. So essentially we had high oxygen and low oxygen crossed with high carbon and low carbon as our experimental factors that we tested. We sampled microinvertebrates as they emerged out of the sediment in the bottles by pouring off the water through a sieve with really fine mesh and catching the microinvertebrates as they emerged out of the sediment. So that ran for a duration of 3 weeks. In the laboratory, we looked at the microinvertebrates under a microscope. So we used a stereo dark field microscope, and we counted and identified the microinvertebrates. Also, the study essentially showed that hypoxic conditions or low oxygen concentrations can reduce the abundance of microinvertebrates in the short term, but returning the low oxygen concentrations back up to high oxygen concentrations actually led to a partial return of microinvertebrate numbers. The exciting thing about the results is that it showed that there is potential for microinvertebrate populations to return after short-term periods of exposure to low oxygen concentrations.
The managers started asking, well, could black water influence the growth and germination of aquatic plants in wetlands? So what we did is we went out and ran some experiments to see whether black water would have an impact on aquatic plants.
So our aim was to take established plants and subject them to different levels of dissolved organic carbon, and to measure the effects, whether it promoted growth or whether it actually had any detrimental effects to their growth. The high levels of carbon turn the water a black colour, and the carbon is actually a sort of basal food source. We were looking at whether or not the plants take that carbon up into their system.
We had one experiment which looked at the germination of aquatic plants. So we went out and collected some dirt, and then we added various concentrations of black water to that dirt and looked at the response of the aquatic plants germinating from seeds. And the other one was looking at how black water would affect the growth of aquatic plants that were already established in a wetland.
We had a replicated mesocosm experiment where we took hundreds of individual plants from 4 different species, and we subjected them to 3 levels of dissolved organic carbon. So we had no carbon added, we had a moderate level of carbon and a high level of carbon. And they were allowed to grow for 12 weeks, where at the end we measured their biomass and total length of growth.
And we weighed the above ground biomass, which is everything above the ground, and we weighed everything that was, or the root mass that was below the ground. We found that in general plants didn't really care about black water. So there was no benefit of having black water going over it. There was no disbenefit of black water going over it. They would still manage to grow quite well. You could almost predict that if you had more carbon you might get more growth, but in that case that didn't happen. It didn't appear to stop any light penetration coming down, so even these submerged plants still managed to grow.
It is a good thing because black water has quite a stigma attached to it that it's a bad thing, and what we wanted to see was whether or not it really affected the plant communities and the ability for the plant to grow, and we showed that even though it didn't have a positive effect, it also didn't negatively affect them either.
I looked after the river side of the project, and what we were really trying to do there is, we know that a lot of materials come off a floodplain, a lot of carbon and a lot of nutrients and a lot of fuel for a river system, but no one's really ever shown how good that material is or whether it has much of a benefit. So our project really wanted to show that a lot of those materials that come off a floodplain actually are really beneficial, and that they're not actually the problem that people necessarily always think that they might be. If you water at the right time of the year or at other times of the year, that material actually has a really strong benefit to the river and not the problem that people just associate with the black water. By being able to show that this process actually happening, it was really useful for us as far as researchers go, because we can now tell managers who are putting this water onto a floodplain, or people who are actually in control of moving water around, that sure, you've got to look after the times when there could be problems associated with that watering, but don't concentrate only on that there are other times when really that's really important, and that this carbon and energy and nutrients could be a really important part of what is actually missing in the river. So as part of what we did in our component, we not only just measured the end product and the actual animals that we're trying to track where the food is going through, but we actually looked at every component of the system. So we were able to use chemical techniques and instruments in the laboratory to measure what sort of carbon and what sort of energy is coming from certain places. We actually looked at the microbes that were carrying out these processes. We looked at the algae and the bacteria that are growing on the slime and the films over the surfaces, right through to the different animals that were ultimately eating the final material. We made up our own special, we call them rigs, and we attached red gum logs to some frames and we put them out in the river. So really what we were doing was making artificial snags, artificial systems in the river. We allowed the things to grow on those blocks of river red gum. We could take them back to the laboratory and then carry out all our various analyses and sampling from those and looking what was growing inside the films that grow over the surfaces of those wooden blocks. We also carried out some lab experiments and some experiments in tanks where we didn't use red gum blocks, but we used smaller surfaces where we could actually control the experiments in a lot more careful ways. For our component of the project, we actually wanted to demonstrate that all of those good things that are coming out of that, out of the floodplain and in those waters, they're actually consumed by a whole range of microbes and microorganisms that are in the river. Then those microbes become food for small animals. Those small animals become food for bigger animals, and ultimately that material ends up being fish food. Is essentially at the end point. So it's really looking at how food and carbon and energy can come off a floodplain and end up inside the larger body fish that actually are out there in the river.